Today my lecture is on resting membrane potential. We know our cells are having a fluid, we call it intercellular fluid. Okay? The fluid within the cell, intercellular fluid. And outside the cell, there is another fluid, and that is extracellular fluid. Now, the composition of these two fluids, particularly the ionic composition, are not alike. They are different. Particularly near the surface, either it is internal surface or at the external surface. So there are ions, and these ions are having different concentrations. The concentration of sodium is not equal to that of concentration of sodium inside the cell. Similarly, potassium ion concentration is not equal across the membrane. In some part, within the cell, its concentration is more, and outside the cell, it is less. So we can see, if we go through the composition table, particularly of ions, we will find there is unequal distribution of ions across the membrane. We will discuss today how this unequal distribution of ions actually made, and what is the purpose of such unequal distribution. Obviously, since our topic is resting membrane potential, so its purpose is to generate resting membrane potential, to create the resting membrane potential. And we will be knowing this potential is very much important. We are living, and one of the characteristics of living cell is there. These cells, all the living cells, are having a potential difference across the cell membrane. This potential difference across the cell membrane during the resting state of the cell, there is a potential difference, and this potential difference is called resting membrane potential. The potential difference across the membrane at rest, it is called resting membrane potential. There are other terms given to it. We will come into this. Come. You know, the life on earth initiated at ocean, sea. The water in ocean and sea is salty. So this salty environment actually favored the initiation of life. Interestingly, our cells are also in our environment, salty environment. Our extracellular fluid is also salty. Definitely, it's got a purpose. We'll come into this. So, for the purpose of your exam, from this topic, resting membrane potential and action potential. These are the things you must know. There may be questions concerning MCQ, short notes, short question, may be based on this. These are the important aspects of this topic, resting membrane potential, and thereafter we'll be learning about action potential. So all this thing will cover this thing. What are these? We'll be learning diffusion potential, equilibrium potential, resting membrane potential, how the RMP, resting membrane potential, is generated. We'll be learning what is action potential, how action potential is generated, and what is Gibbs-Dunan membrane equilibrium, Gibbs-Dunan phenomena, and its effect, Dunan effect, and Nernst equation. What we do actually with a Nernst equation, what can be derived from this equation. There is another equation, Goldman-Hodgkin-Cutts equation. And what is this equation is for? So we'll be discussing. And role of sodium and potassium ATPase, so that is sodium potassium pump, in genesis of resting membrane potential and maintenance of resting membrane potential. RMP, we will be studying RMP, resting membrane potential, is not same and not identical in all tissues. It varies in epithelial tissue, in RBC, in muscular tissue, in cardiac muscle, in nerves. Okay, it varies. So among all these cells, there are some other type of cells. We call them, other type of tissues, we call them excitable tissues. What? Excitable tissues. Can you name some excitable tissues? Yes, neuron and muscle. It may be cardiac muscle, it may be smooth muscle, it may be skeletal muscle. So they are also having resting membrane potential, and since they are excitable, so it is also very much important on resting membrane potential. And this will determine the excitability of the tissue, of such tissues. How easily you can make them excited. It will depend on the level of resting membrane potential. Okay, so this is important. We will see this is different. For nerves can be stimulated easily. And the cardiac muscle cell, stimulation of cardiac is not so easy. It will be difficult, or strength of stimulus should be, it will require more strength to excite them. And it is because of this resting membrane potential. So, we will see different ions. Different ions are contributing the generation of resting membrane potential. So there are sodium ion, there are potassium ion, there are chloride ion. Which ions? There are protein ions staying within equilibrium potential for potassium ion, sodium ion. So we have to study this thing. You see the distribution of ions inside the cell and outside the cell. You just go through the sodium ion, potassium ion, their level inside and outside. Chloride ion inside and outside. These are the main ions, and the protein ions inside and outside, because they are the contributing factors. They are responsible, these ions are responsible for generation of resting membrane potential. So you try to remember this. This is also from another book. So somewhere it is sometimes a little bit difference is there. So it is the gist. So main ion, this main ions concerned with this resting membrane potential or generation action potential are this. We'll come later. I will come later into this. These are what Dunant effect, Gibbs-Dunant phenomena, and Gibbs-Dunant effect. Now, the description of what our discussion, and this we will limit our discussion here. Cell membrane, we will see that cell membrane is not just a boundary. A potential difference, as we said, exists across the membrane, and difference in electric charge between outside and inside of a membrane, we will see this, and inside of the membrane is negative in respect to the outside, and the difference is called the membrane potential. We could also, just now we learned, and membrane potential exists in excitable and non-excitable tissues, already been told. And you see, what is this? What is this? In your physics lab, you have used it. Voltmeter, correct. Voltmeter. What we do with voltmeter? We find the voltage difference. Voltage. When there is difference in polarity in two sides, then what is the voltage difference? You will find in ECG, while recording ECG, there will be voltage difference here to here, here to there. Okay, this hand to that hand. We will find there are potential differences, and this is recorded in electrocardiograph machines. Okay, so how it is, then we will discuss. So this is important. This is by voltage difference. We record with this voltmeter, and modern use is using another equipment. Instead of voltmeter, we use it CRO, cathode ray oscilloscope. Cathode ray oscilloscope. So using cathode ray oscilloscope, we can very nicely get what happens during action potential. What are the changes actually made when action potential is generated, when action for every action. See, I contracted my muscle, so I have to generate action potential, and that action potential travel through nerve to the muscle. So there, for nerve is not directly attached to the muscle. There is a junction. We call it neuromuscular junction. So this impulse action potential ends at the junction, and here it causes liberation of a transmitter. Okay, it may be acetylcholine, it may be epinephrine, okay, it may be gamma aminobutyric acid, depends what is going to do. What is going to do to its target organ. So the muscle, in the muscle, it will produce again action potential, or in the next neuron, it will produce again action potential, and this action potential will be traveling along the cell membrane, either of neuron or of muscle. What is the name of the muscle cell membrane? Muscle cell membrane, what is its name? Yes, sarcolemma. And the cytoplasm within the muscle cell? Sarcoplasm. Good. And the sarcolemma, so this will be traveling through sarcolemma, and the sarcolemma is inserting into the cell through a tubular structure. We call it sarcotubular system. So at the end of the circular system, there is a sac-like structure where calcium remains stored. So when this action potentials will be traveling along the muscle cell wall, I mean not wall, membrane, sarcolemma, it will penetrate into the cell through sarcotubular system, and when it reaches the sac, from there it will liberate calcium ion. And this calcium ion will bind with actin and myosin head, and here it will also attached with ATP. It will be broken down, energy will be taken. With that energy, actin and myosin will be sliding and will cause contraction. So this is the process. So for this purpose, we need action potential. Without action potential, it cannot be done. Without action potential, nerve cannot be transferred from one cell to other. So generation of action potential, not only this, the nerves are controlling, some nerves are controlling our glands, digestive glands. So the efferent nerve that is coming to the gland, so initially it should come as an action potential, and there it will stimulate, then only it stimulate the gland for secretion of the gland. Okay, and this secretion will perform certain functions. Okay, so for that purpose also action potential needed. So it is important. We could say it is important for carrying the sensation to the brain. For we are having different type of receptors in our eyes, in our ear, on the tongue, in our skin. So all these receptors, unless stimulated, this sensation will not go to the brain. And for stimulation means this receptor means there should be generation of action potential. Then it will be transferred to the brain, and we will feel something has happened in the periphery. So in this way, sensation is coming. Then the brain will decide what to do. So then it will produce motor action, or then it will be going through another nerves, motor nerves, to the periphery, or to the efferent nerve, to the efferent. Efferent is carrying, efferent are delivering. Okay, so all these things will involve action potential. So this is very important. So this is, you see, what is this? This is a cell, this is the membrane. As we have seen the composition of body fluids, we have seen the composition that outside of the cell there is more sodium ion concentration and they are positively charged. Okay, so that's why outside is positively charged, whereas inside is negatively charged, because there is though potassium ion is more, but in relation to that negative ions, that is protein, chloride, sulfates, their negativity is much more in relation to the exterior. Okay, so because of this, outside is positively charged and inside is negatively charged. And now we put two electrodes, one at the surface of the cell, another electrode on the inner surface of the cell, and connect it to a galvanometer or CRO, cathode ray oscilloscope. We'll find what? This is the 0 millivolt. Okay, this is the pointer. This pointer deflects to the negative side. It means the difference across the membrane is minus 70 or minus 80 or minus 90 millivolt. It is minus. Under resting condition, it is minus. It may vary 40 to 90 millivolt depending on the type of tissues. Understood? So, this is one electrode is placed on the surface, external surface, and another electrode is placed there. And the cell is under resting condition. So, here we find a negative deflection, and this negative deflection shows how much negative is inside in respect to the outside. And this is recorded to be, say for nerve fiber, it is 70 millivolt. Okay, so it is the resting membrane potential and how it is determined. So this is the way how we determined or come to know the resting membrane potential of different tissues. So this, as I said, it has got other name also. What is this? Another name? Another name? Another name, transmembrane potential. So don't be confused. If it comes, so this it is called a potential difference noted during the resting condition is called action potential, is called resting membrane potential, is called transmembrane potential. Say depolarization potential. So it may be written like that. So you have to choose which two are same. So you will find transmembrane potential and resting potential are actually same. When we see, you see the table, this table, or this one, is this side polarized? Are there any charge in this side, other side? So if you connect through a galvanometer, you won't find any difference. But whenever we'll find there is charge, one side is negative and another side positive, then we'll call it is polarized. Then it will call, there is cell we buy from the market for different gadgets. Okay, so this says one end it is marked there, one side is positive, so it is a polarized. It is polarized. Similarly, our heart, it is also polarized, and it is always with every beat. There is electrical changes in the heart, on the heart surface of the heart. So sometimes it is some area is positive, sometimes that area is negative, and depending on this, the ECG waves will vary. Okay, so now since the membrane at one side there is one charge and at the other side there is another types of two different charges are there. So we will call it in this condition, you will call it is in polarized state. It is in polarized state. It may happen. Now you see this. Is it in polarized state? Is it in polarized state? Yes. Because outside is positive and inside is negative, there is, because of this positivity outside and negativity inside, there should be a potential difference. When it happens like this, it shows potential, that means it is polarized. So this cell under membrane is in polarized state. So when there is RMP, resting membrane, so we know the membrane is in polarized state. Now it may happen, positivity may become inside and the outside may become negative. Can we say it is now polarized? We will now term it as depolarized. This doesn't happen in normal condition. It happens in generation of action potential. It happens during action potential. Then only when outside became negative, inside become positive, and we'll see how it has been done. It is done actually. how this is reversed, how it is depolarized. And when this depolarization comes to a particular level, then only action potential is generated. So, for action potential, this membrane has to be depolarized. Okay, so when it is depolarized, then this will travel along the membrane. When it will be proceeding, when it will be proceeding, leaving this space, coming to new area, the area it left behind, it will be simultaneously coming back to its normal position. Means in the polarized state. There will be again reversal. Means outside will become positive, and this is called repolarization. Repolarization. Again it was disturbed, now again it came back to its original position, so that next time I can stimulate it. Otherwise, once my muscle is contracted, again if it is not relaxed, if the resting membrane potential of the cell is not brought to the normal level, it cannot be stimulated again. So repolarization is a must. Depolarization is essential for generation of action potential. Repolarization will help to return back to the normality. So these are also very important. Already told, and this is actually diagrammatic image of crow, and the variation among different tissues, variation of RMP. You see in some tissues, in nerve it is 70 millivolt, as I said, and in skeletal muscle in 90 volt. Question may come. Question may come means MCQ may come. So smooth muscle is having 35 to 45 millivolt. What is the use of this resting membrane potential for smooth muscles? In our GI tract, one muscle coat is there. One muscle coat, if you take a section, so you'll get at the center between mucous coat and external coat, in between there is a muscular coat, and these are made up of smooth muscles. So what are the use of the smooth muscles? What they will do in my GI tract? What happens when you eat some food? It will go through esophagus. So you have to swallow it. For swallowing the food through the esophagus has to be contracted and relaxed. So as a consequence of this, it will be traveling. When the food enters into the stomach, my stomach will start contracting, relaxing frequently. Purpose? Churning, mixing with digestive juices. So who will do it? The muscle of the stomach. And for this function, for this purpose, contraction of the smooth muscle, action potential is needed. So smooth muscle must have also resting membrane potential, isn't it? Otherwise, it is the movement of the GI tract, peristalsis, it will not happen. Now the genesis, how, as I said, how negativity is developed inside, how this happened. So we'll discuss it, and what are the reasons? These are the reasons by which, these are the factors by which we can explain how RMP is actually generated. How it is developed. Okay, what are these? Differential permeability of cell membrane. we know that our cell membrane is semi-permeable. Means what? It allows somebody to pass through it, and for someone it is impermeable. So it is for this nature of this. One factor is this. Another factor is the ions. Some ions are like elephant and some ions are like rat. See the size. So whose size is big but the door size is small. So it is not able to pass. One example, the proteins that are present in our cell. The proteins that are present within the cell, they cannot pass through. They don't have any gate, any port, or any, they don't have, for them, no carrier, no channel who can transport these proteins to the exterior. Okay? So they will remain inside and will be negatively charged, remaining negative always. And there are some other ions too, like protein, sulfates. These sulfates are there within the cell and its concentration is higher than exterior. Within the cell it is more. So they have also negativity. How many negative charges for each? SO 4, 2. So they are having more negativity. Every ion is having more negativity. So they are remaining inside. And this is the differential permeability and differential distribution of ions across membrane, and we'll see how it happens, differential distribution. One is the protein. It cannot live. So it has to live within the cell. So there will be differential distribution. Outside there is no such protein. This amount of protein there is absent. So there will be unequal distribution of this. And there will be unequal distribution of not only this, there will be unequal distribution of sodium, there will be unequal distribution of potassium, chloride, but so they will be unequally magnesium ion, chloride, calcium ion, they will also be unequally distributed. So out of these, what is the contribution? Sodium, potassium, ATPase pump. Are there any role of this in generation of negativity inside? We'll see. Yes, it is there. It has got some role. And next is Nernst equation. With this equation, by using this equation, we will find the role of each ion, role of sodium ion, how much you can generate. If you work alone without the support of sodium, other ions, if other ions are impermeable, only you are permeable to pass through the membrane, then how much RMP you can generate? So we can calculate it by using Nernst spelling N-E-R-N, then ST. How you will pronounce it? Nernst equation. Okay, so with this Nernst equation, why you will be using it? To know the contribution of different ions like sodium, potassium, and separately, what is their role? How much they can generate? What is their contribution? That we can measure, we can estimate. From the inside and outside their concentration will be there. By using this concentration, how can we derive the potential difference? Okay, so for that purpose, Nernst equation is used. And another equation there, what is there? Goldman-Hodgkin-Kurtz equation. Learn this? Okay. So we told you the purpose of Nernst equation. Now, what is the purpose of the last equation? Nernst equation, on the sodium, his contribution you came to know from Nernst equation. For sodium, that too you came to know how she has performed in generation of RMP. Her contribution we came to know from Nernst equation, chloride from Nernst equation. But it's not that all are acting together. So, while they will be acting together, sodium ion, it will act in its way. Potassium ion will act in its way. Other ion will also. So, in combined effect, how much potential will be generated? That can be estimated using the last equation. So, in your how this equation is derived, Nernst equation or Goldwyn, this type of question will not come. What will come? The utility, usefulness, purpose, what it says, what information we can get from this. And the last equation, you can just mention, this is the equation. How it has been derived by the mathematician, that is not required. Okay, only you have to explain, you have to mention what actually this equation, what information it provides. So these are the saying, and the requirement of already tell you. Another thing is there, Gibbs-Donnan equilibrium. Gibbs-Donnan equilibrium. We have seen what is the effect of Gibbs-Donnan effect. What is this? This equilibrium, Gibbs-Donnan phenomena. What is this phenomena? This phenomena is side A, side B, and there is a semi-permeable membrane separating the both solution. Semi-permeable membrane. Okay? So I put in A potassium chloride. Here also I put some potassium and chloride. Semi-permeable membrane, both can pass along. No issue. Nobody is going to obstruct it. So what will happen? From higher, if concentration of potassium ion is higher in A, then if its concentration is higher, sodium chloride is higher here, so what will happen? Potassium ion, here it is less, there is a concentration gradient. This is higher, this is lower. So, potassium ion will move from higher to lower because of concentration gradient. So, this will move there, and similarly, it has to be high again because potassium and chloride, positively charged, negative, they will like to stay together. So since it has gone and concentration is also higher, here it is less, so chloride ion will also move there, and after some time what will happen? Both sides will become to a concentration. It means concentration. We'll say. So concentration of this will be this. Concentration of this will be equal to this, and concentration of this will be equal to, not concentration, charge. How many charges is there? Positive charge? Okay, 5. How many charges are there? Okay, 5. So these are neutral. No charge difference. Electrically neutral. So that side will also be electrically. Now it is electrically neutral. As well as the concentration of this, since the concentration of this and this are equal, since the concentration of this and this are equal, so concentration of this and this will, not concentration, charges will also be equal. Okay. Now, so you see distributed equally, both sides, equally, no difference in concentration or in charges. Now, see what happens. Now, see what happens. I replace this value with PR negative, means protein. Now, this protein is not permeable. Now, this protein is not permeable. So, what will happen during equilibrium? Equilibrium will state will come. So, what will happen? We will see that more potassium ion is accumulated here because of the presence of non-diffusible ion. Here its concentration will be less. Concentration of chloride, since it is moving, this one will also be moving, and the product, the previous equation, KCl, this side, KCl, this side, okay, their product, this into this into this should be equal. Their ratio should be equal. But here neither the product is equal, neither their, since these are coming, so its amount will be less here, its amount will be less here, but its amount here it will rise because of the presence of this. There will be unequal distribution of ions now. Only because of what? Because of presence of protein, which is non-diffusible. So we can see now, if one side contains a non-diffusible ion and if it is positively charged, so at this side means here it is a side A, so at that side the cations means the positively charged ions, cations means and cations, anions, okay. So these are what? Anion, and these are cation. So this is impermeable. So whenever we find there is an impermeable ion, so in that ion there will be level of potassium ion, means cation will also be compared to higher. Compared to B will be higher. So as a consequence, there will be what? Chloride also higher at this side. So as a consequence, what will be there? Unequal distribution of ions. So see, presence of one impermeable ion in one side affected the distribution of ions along the body. So this Gibbs-Dunan effect you also will see there. Now you go through the Gibbs-Dunan I have written there. You now study this. Read it. Read this. Try to understand this. Presence of non-diffusible ion on one side of the membrane affects the distribution of other ions to which membrane is permeable. So only this will affect the permeable ions. It happens in case of cations, it also happens in case of anions. Okay, and this results in asymmetrical distribution of diffusible ions and also total ions across the cell membrane. So who is the responsible? Gibbs-Dunant effect. And it is because of, this is called Gibbs-Dunant effect, and it is because of existence of impermeable ion within the cell. Mainly it happens within the cell. Gibbs-Dunant phenomena. What is this? Now we also go through. It is due to presence of impermeable anions like protein sulfates in one side of cell membrane, that is inside. Many negatively charged ions are present inside the cell that cannot pass through the membrane. They cannot pass through. They are anions, organic substances, anions of organic substances such as protein and sulfates. Because of their impermeability, the interior of the cell becomes more negative in respect to outside of the cell. One of the cause. Why the inside of the cell is negative? You have to, if it is asked, so you have to write this. This is the cause, one of the cause. There are some other causes. This is one of the cause. This is Gibbs-Dunant effect phenomena. There is net deficit of diffusible cations. This is called Gibbs-Dunant phenomena. Okay, and Gibbs-Dunant effect. What is the effect? Asymmetrical distribution of total ions across. Only same thing. Asymmetrical distribution of total ions across the cell membrane at equilibrium has the following effects. There will be, and since there is, so ions are unequally distributed, so there may be unequal distribution of ions too. So there will be generation of electrical difference, potential difference, and this will cause a membrane potential, generation of membrane potential. Okay. See the next part. Because of presence of more anions, that is protein, inside the cell, then what will happen? It will start accumulating substances that will create more osmotic pressure. If it exerts more osmotic pressure, then water will be entering from outside into the cell. As a result, cell will be increased inside and it will burst. But it practically doesn't happen. Why? Because there is one pump. This will happen because of entry of the sodium ion. So if the pump, sodium-potassium pump, pumps out sodium outside, then the osmolarity will be reduced, so there will not be accumulation of water, so the cell will not die. But what will happen? If I add some drug that will collapse my sodium-potassium ATPase pump, what will happen to the cell? Sodium that has entered will not be eliminated, will not be sent back to the exterior. There will be accumulation of sodium. So in addition to other osmotic pressure creating substances, sodium will also be adding. So they will increase the osmotic pressure. As a result, it will draw water. Osmosis will be there. So from outside, water will be coming since it is higher in concentration here. So the cell will be inflated and it will die. So this is the effect, Dunant effect. Now we will discuss about the role of sodium-potassium pump. I'll take this in practical class. There is 3 hours time, no? So 1 hour time I will take for this purpose. I'll finish action potential and the rest part in the practical class. So only 50 students will be there. So this will be taught in 3 classes. 1 class for A, 1 class for B, 1 class for C. This is very important for you. So you must understand. And here too, these are related to when will be muscle contraction, nerve muscle physiology. It is also important. In while we will be studying nervous system, there also it is very important. So it is to be clear in your mind. The concept should be very clear to you. There should not be any doubt. Okay.